Well, this is super exciting to announce the inaugural Brain Body Lecture Series. This will be a monthly series, so please tune in month after month and join us for an outstanding lineup of speakers. For this first talk, I'm very proud to have a fitting and I think expert in this area of brain-body research. So I just want to give this like very brief introduction because I don't, Phil and I met probably for the first time this past year, and it's he's kind of like the other half of me intellectually, basically. I'm a neuroscientist retrained in immunology. He's the immunologist retrained in neuroscience. So we immediately sparked up, I think, a really great collaborative effort, a friendship, and I can't think of anybody better to kick off this inaugural lecture. Phil's lab is just a powerhouse. I don't have to tell you guys, it's just been a pleasure to see him come here, build an amazing group, and then, month after month, publish a Nature paper. And so he's both an impressive leader, but also probably one of the best scientists that I've known. So I think what he's going to talk to you about is some of his recent data where he's defined these very discrete circuits starting from stress centers in the brain and then emanating out into the periphery, and how those stress centers can control immune function and then ultimately health or disease-like states. So I'm going to let Phil tell you more about his research, but again, I'm very proud to have Phil as our inaugural speaker. I'm very proud to have this lecture series. Please go to our website and check out the rest of the lineup for the year. It's exceptional. We're going to continue into 2023. We'll remain a hybrid format so that both within Mount Sinai and outside of Mount Sinai, we can promote brain-body research. One more little bit of housekeeping for questions online. I will field them, so I can kind of call you out, and then you can unmute yourself and ask your question directly out into cyberspace, and then Phil will hopefully address that. But without further ado, let's give a warm welcome, please, for Dr. Phil Swirski.
Thank you, Scott, for this introduction. It's a real pleasure to be here with you physically and those who are joining virtually. I think this Brain Body Center is really fantastic, super timely, and this is a very important area of work, and I'm very happy to be collaborating with Scott. I think there is some really amazing science that we are poised to do here. So what I want to do, I'm relatively new to Sinai. I started just over a year ago. The lab, the entire lab sort of moved from Boston, and so what I want to do today is first give you a little bit of context in terms of what are the questions that we're asking and why are we asking the questions, and then share with you some data, some recently published data, as Scott mentioned, and some data that is not yet published, and sort of two vignettes that very much fall into the themes that I will introduce at the very beginning. So what we've been really interested for a long time is what I like to call systemic inflammatory networks or inter-organ communication. And a lot of this work really has been done in the context of cardiovascular disease. This is where a lot of my interests started as a postdoc and have continued. And the realization here was that cardiovascular disease, whether it's atherosclerosis or sequelae of atherosclerosis, cannot be really understood without considering organ systems. That really there are many organs that contribute to cardiovascular health and its breakdown. And further, that a major communicator of organ system is the leukocyte. So we've really focused on leukocytes as these are immune white blood cells that, of course, participate in the immune system, but as we have found and continue to find, are also integral to body systems. And as motile cells, these cells really connect organs and organ functions. And so in terms of trying to better understand cardiovascular disease, a major place, a major location of interest has been the bone marrow, as this is the birthplace of immune cells, of white blood cells, but also red blood cells. We have been interested in mechanisms that are relevant to leukocyte mobilization out of the bone marrow. We've certainly been very interested in the blood and secondary lymphoid organs such as the spleen. A key site, of course, is the vessel wall, as this is the place where atherosclerotic lesions grow. And then, of course, when atherosclerotic lesions erode or rupture, that can lead to heart attacks or stroke. And so for this reason, the heart has been of interest in the brain. And in the last several, I would say maybe 7 years or so, we've become particularly interested in the brain, not necessarily as a place where strokes occur, but as a place, as a location that interprets pain, but can also do through various systems, can also control much of the sequelae and the immune response that occurs subsequent to myocardial infarction or stroke. And so this is really the broad view, but we want to also dig in a little bit and better understand not just what happens between organs, but what happens inside each organ at the cellular and molecular scale. And for this reason, we continue to be interested in mechanisms that occur in the bone marrow, as again, this is an area where hematopoietic stem progenitor cells must be on the one hand protected from the, let's say, hostile outside environment insofar as they must remain throughout life to give rise to white blood cells and red blood cells. But on the other hand, this is a location that imparts exquisite control over the production of white blood cells and red blood cells, many of which have very different life cycles. So starting from neutrophils, which can live for a day or so, to red blood cells, which can live for 120 days, to some cells that can live for months and months and months. And so there's exquisite control. And the bone marrow is the location, the bone marrow niche, which consists of multiple cells that produce multiple cytokines, chemokines, growth factors, and so on and so forth, that really sustain the proliferation, differentiation, sort of the lifespan of hematopoietic stem cells. Another location that has been of immense interest to us over the years historically is the vessel wall. And we've learned over the years that atherosclerosis is not just a plumbing disorder, it is not just the accumulation of lipids in the vessel wall, but it is also a disease that is driven by the recruitment of leukocytes. And at the risk of oversimplification, for atherosclerosis to be, you need really two things. You need lipids and cells. And you can cure atherosclerosis by essentially trapping monocytes in the bone marrow. So there are certain leukocytes that are very important to atherosclerosis. I would say that all leukocytes have been implicated in the disease one way or another, but monocytes arguably are probably the most important cells in terms of the sheer number that accumulate in a growing lesion and give rise to macrophages that then can proliferate, efferocytose, produce a whole panoply of growth factors, cytokines, and so on that really shape the atherosclerotic lesions. And they really participate at all stages in not just the growth of atherosclerotic lesions, but also their rupture or erosion. And so for this reason, this is continues to be an important area of investigation, especially now in the era of clonal hematopoiesis and with the sort of the realization that the immune system and inflammation in general is really a major component of cardiovascular health and cardiovascular disease. And I also mentioned, what happens when atherosclerotic lesions erupt or erode. This, of course, may lead to myocardial infarction And myocardial infarction, of course, is ischemia, and that leads to profound mobilization of the immune system, and specifically of the inflammatory cascade, resulting in the recruitment of neutrophils and monocytes and other cells that then talk to and interact with some of the resident cells in the infarcted myocardium, participate in the inflammatory phase as well as the subsequent reparative phase, giving rise to a scar. And I would argue, and I think I'm not alone in arguing this, that the nature of this response, both the quality and quantity of the response, has a major effect on whether one develops heart failure. So surgically, we're very good at, or surgeons and sort of healthcare is very good at saving lives acutely. But there is, of course, the question of the scar that may develop, and which may be very important to whether 1 or 2 or 3 years down the line a person develops a cardiomyopathy or heart failure. And so again, this something that was really studied in sort of dusty corners of academia 15, 20 years ago, but it's now really becoming, coming to the forefront of cardiology. And I'm happy to hear that cardiologists are sort of realizing that this is an important area. And I'm certain that this will continue to be an important area in the future. And already there has been evidence that sort of targeting some of these pathways can really save lives. So that's just the background and really sort of the context, and just keep that in mind as I continue this talk. What I want to focus now is something that we've become very interested in for the last sort of 7, 8 years. I don't know how many now. I used to say 4 years, 5 years, and every year have to add a year. But it's been a while. And that is the role of lifestyle factors on cardiovascular health and disease. And the question is, why lifestyle factors? So we know that genetics play a role in cardiovascular health. There's no question about this. But of course, we know that there are specific risk factors. The Framingham Heart study and the various risk factors that we know are essential to considering cardiovascular health, whether it's cholesterol levels, hypertension, and so on and so forth. And when you look at some of these risk factors and you just take them to the logical conclusion, many of them really can be traced directly to lifestyle factors, to the environments that we inhabit voluntarily or involuntarily, and to the lifestyles that we engage in throughout life. And so the question here was really to study those lifestyle factors and better understand how they impact some of these pathways, these tissue, organ, and molecular pathways that I already introduced. And the idea here is not to prove or disprove whether a certain lifestyle is good or bad for you. I think we can all understand that we should be exercising, we should be sleeping, we should be avoiding certain kind of stress. The idea is that by studying lifestyle factors in their entirety, so in other words, sleep as something that we do and should be doing and engaging in for a third of our lives rather than the circadian clock, that we might identify pathways that perhaps we were unaware of that can help us better understand how the body adapts to external stressors. And then perhaps those pathways can then be harnessed on the one hand to engage with public policy and change perceptions of certain of how lifestyles keep us healthy, and on the other hand perhaps even develop drugs that engage those pathways to give us a helping hand and extend our lives, or at least make them extend the quality of our lives. If we can't live forever, at least maybe we can live better lives, into our sort of sunset years, so to speak. So this, so these here are 4 lifestyle factors that we've been particularly interested in. They're by no means exhaustive, but ones that we have done some work in. And I will, in this talk, just focus on 2 of them. And perhaps in some time in the future, I will share with you some of the others, which is ongoing work in the lab by and at different levels of different stages of development. So first, I want to talk to you a little bit about sleep. And of course, sleep is something that we all know about, we all care about. And again, this is not to convince you that sleep is important, but just by some numbers, we know that sleep deprivation or obstructive sleep apnea elevate risk for cardiovascular disease such as atherosclerosis, myocardial infarction, and so on. And we also know that we are not getting enough sleep, and this is a problem that has been identified not only in the United States, but the world over. And so this is a lot of the work on sleep has actually been done by a former postdoc of mine, Cameron McAlpin, who's here in the audience, who is now an assistant professor here at Sinai, is continuing in this work. So I'm just going to summarize some of this work here in this slide and 3 more slides. So the idea here is to better understand how sleep affects the hematopoietic, immune, and vascular system in general. And so in order to do so, we really need to use the tools of neuroscience, we need to use the tools of immunology, we need to use the tools of vascular biology. So we need to be able to profile sleep patterns and really connect the dots, so to speak, between what happens in the brain and what happens in this extremely important location, the bone marrow, the birthplace of immune cells and red blood cells, and then connect that again to the vasculature, and what happens in the context of atherosclerosis and its consequences. So this is really a sort of a broad scheme, that a map or a blueprint, if you will, that helps guide the experiments that we seek and that we do. And so in work that Cameron published as a postdoc in Boston now 3 years ago, really asking the question, how does sleep, or first of all, does sleep do anything to cardiovascular disease? And if so, how does sleep control some of the processes that we care about? So I will only summarize this in this slide here because, of course, this is now a 3-year-old paper. But the idea here is that atherosclerotic lesions, as I mentioned already, grow via the recruitment of immune cells, via the recruitment of monocytes and the accumulation of lipid. And we know that monocytes continuously accumulate in the mouse. There are these Ly6C high monocytes. There is an equivalent monocyte subset in humans. And these cells are made in the bone marrow. And so what Cameron discovered here was that there is a subset of pre-neutrophils. So these are immature neutrophils that reside strategically in locations where hematopoiesis is very active. And these pre-neutrophils produce CSF. So this is colony stimulating factor 1. So this is not cerebrospinal fluid for the neuroscientists in the room. So they are not the only producers of CSF. Other cells produce it, but they produce enough of it to make a difference. And what makes these pre-neutrophils doubly interesting is that they express a receptor for hypocretin, which you may also know as orexin, which is produced in the hypothalamus. Now, this is a wake-promoting and appetite-promoting neuropeptide, the cause of the destruction of which is the cause of narcolepsy. And so, as we're awake, hypocretin levels are up. And what Cameron discovered here is that hypocretin, via the hypocretin receptor on neutrophils, acts as a sort of brake on the bioavailability or the production of CSF1. So what that in essence means is that when, in this case, mice are awake, hypocretin levels are up, and that limits the amount of CSF1 that is produced, thus limiting the number of monocytes that are being produced by the bone marrow. Now, why would that happen is something that we can discuss. We think it's about energy allocation, but it's really a cyclical circadian pattern. So when the mice are asleep, hypocretin levels are low, and that increases CSF1 production and then increases the production of monocytes, which then are recruited to the vessel wall, which can propagate atherosclerosis. Under conditions of sleep fragmentation, and there are some other sort of sleep influencers that you see on the right there, that system is off balance. So the levels of hypocretin over time chronically begin to be impaired. There's less hypocretin that is produced. And as a consequence, the brakes are off, so to speak. More CSF1 means more monocytes. More monocytes means more atherosclerosis, especially under these conditions of hyperlipidemia. So this was the insight of this paper. And of course, any paper should, answer certain questions, but certainly raises sometimes even more questions than it answers. And so there's a number of questions that immediately may jump out at you here. One of which is, is this relevant in human? Because this was a mouse study. Another one of which was, well, what happens in the long term? How permanent is this phenomenon? What happens if the mice are allowed to sleep? Is it abandon all hope, ye who enter here, as it says on Dante's Inferno, or is there hope in terms of this system really responding and setting itself back when sleep returns to normal? So this was really the point of a subsequent study, which is now in revision, also led by Cameron. The human component of this was done in collaboration with Marie-Pierre Saint-Onge at Columbia. This was a very nice sleep restriction study, a crossover study using objective criteria of sleep analysis with actigraphy, with essentially people who sleep normally versus those whose sleep is restricted for an hour and a half each day. And then what was collected were sort of immune cells from the blood. And so sleep duration was indeed reduced in those undergoing sleep restriction. And those undergoing sleep restriction, we found, had fewer monocytes, or at least, more monocytes in their blood in the evening. And this is actually very important. So there were no changes in the morning, but there were changes in the evening. And this has to do to the circadian fluctuations of monocytes in the blood, and very much reflects what we saw in the mouse. And it's important to consider because a lot of blood that is often collected in humans is collected in the morning, and if you just do this in the morning, you would never see any differences, but the differences really arise in the evening. And this has something to do with this concept of anticipatory inflammation and the flux of cells between the blood and the peripheral tissues. But this reflected and was in many ways a phenocopy of what we saw in the mouse. So we thought that this is exciting. We don't know whether this is based, it has something to do with orexin, but at least tells us that perhaps mouse work is not useless after all, and it can reveal some mechanisms that then can be transferred to the human. Keeping in mind that the circadian clock was also discovered in animals, and in animals much, quote, simpler than the mice. So there is use to animal studies. How long-lasting are these effects? So one can sort of ask this question in two ways. One which is, well, how long-lasting is the effect of sleep disruption on sleep itself? And then, of course, one can ask how long-lasting is the effect of sleep disruption on that hematopoietic system that we saw as disrupted, and specifically on the production of monocytes. So first, using telemetry and really enumerating wake bouts and slow wave sleep and so on and so forth, what we find is that when mice sleep is disrupted when they want to sleep during the sort of the dark phase, if you will, during the day, but during their rest phase, they sleep in. So you can see that here. So they sleep in a little bit because their sleep is disrupted. So once their sleep is no longer disrupted and they are allowed to sleep, then certainly they take a little nap in the morning or in their morning. And it also takes a while, if you let them sleep normally, for their sleep to return to normal. In other words, the sleep disruption that occurs for 16 weeks really has an effect on the capacity to sleep well. But eventually, their sleeping time returns. as you can hear, to sort of to normal in terms of their wakefulness and their sleep patterns. So that's what happens with sleep. What happens with immune cells? So one way to look at this is, and I will just show you sort of one or two experiments that I think drive the point across, is to rely on this competitive adoptive transfer. So this is a classic experiment in order to look at what happens in cells versus what happens in the environment that they dwell in. And so here there was an experiment that involves the adoptive transfer of LSKs. So these are hematopoietic stem progenitor cells that will give rise to neutrophils and monocytes, from animals that were controlled, these CD45.1, so these are animals that are allowed to sleep, versus animals that have undergone sleep fragmentation for a period of time. And then you essentially combine these cells one-to-one, and then you adoptively transfer them to a GFP mouse. And the GFP here is meant just to distinguish the cells that you've adoptively transferred from the endogenous cells, because otherwise the GFP are 45.2. And when you do this, what we find is that the cells that were derived from the mice undergoing sleep fragmentation proliferate at a much higher rate. As you can see here, using BRDU. BRDU is a nucleotide analog that incorporates into dividing cells. And this gives rise to a larger population of monocytes. So there is this competition here, and it is these cells that have been in mice undergoing sleep fragmentation that proliferate at a higher frequency and give rise to a larger population of the monocytes, which speaks to the idea that whatever effects are happening are intrinsic to the HSPCs. So these LSKs are now in an environment where the mice sleep normally, and yet they can still proliferate at a higher rate and give rise to more monocytes. This, of course, raises the question, what's going on here mechanistically? And what I can tell you is that this has something to do with the epigenome of these LSKs. And I won't dwell on this, but there is enhancer regions that are changed, and even after 10 weeks of so-called recovery sleep, there's still an imprint, an epigenetic imprint that we think is the reason why these LSKs give rise to more monocytes and neutrophils. And this work is in revision, again, led by Cameron, that we hope to see hopefully soon in print. All right. Just a little bit of a shout out. This is kind of a little bit out of the blue, but I wanted to show this one slide. What I mean by the blue is that it doesn't have anything to do with lifestyle factors or cardiovascular disease, but it was a study that was published earlier last year on the role of interleukin-3 and the communication between astrocytes and microglia in Alzheimer's disease. And this is an area, sort of while this study really showed communication pathway between astrocytes and microglia in Alzheimer's disease, this really sets the stage for the questions we are interested in pertaining to lifestyle and specifically and better understanding how lifestyle factors may affect the progression, the onset and progression of Alzheimer's disease. So I hope to show you more of this in the future. And again, this work continues in Cameron's lab and in our lab as well. All right. So let me now, in the last 20 or so minutes, tell you a story about stress. So this is a story led by Wolfram Poller, who is an instructor in the lab here. A lot of this was done in Boston, but a lot of it was also done here in Sinai. So it's a product of both of those institutions in that respect. It is a study that was just very recently published, so I think I can still talk about it, but probably this is the last time I'm going to be talking about it as we shift to the new unpublished work. So this story is about better understanding how stress affects the hematopoietic system. And of course, stress is very complicated. And the more one thinks about stress, the more complicated it seems. There is, of course, acute stress. There's chronic stress. There's different types of stressors. Stress is, of course, necessary to protect us against danger, but it can be pathogenic under certain conditions. What Wolfram, when he came to my lab in Boston as a postdoc, what he wanted to understand is how does acute stress affect the hematopoietic system? Or I guess the first question was, does acute stress affect the hematopoietic system? And if so, how does this happen? And it really, what I want to do first is I want to basically tell you the punchline. I'm going to skip right to the end and tell you what it is that Wolfram found in his in this project, and then I will share with you some of the data in support of this conclusion. Okay, so the first question really was, okay, so stress is somehow perceived in the brain, and so how do we go from the perception of stress in the brain to something that happens in the periphery? And here I'm illustrating sort of the atherosclerotic lesion in the heart, which was actually not specifically in that paper, but it's still something that is very much near and dear to how we think about all of this. So how do we connect the dots from the perception of stress to what happens in the periphery somewhere in various organs and among various leukocytes? And so what Wolfram found is that there are certain regions in the brain that have a major effect on what happens in the secondary lymphoid organs. So this is not a kidney, this is a lymph node. And so certain regions of the brain empty out, if you will, maybe not entirely, but have a major impact on lymphocytes and monocytes that otherwise inhabit these lymphoid structures, which are very important to the acquisition of adaptive immunity. And so they induce mass scale transfer of leukocytes from peripheral organs, and importantly, in this case, from secondary lymphoid organs to the bone marrow, to that region where hematopoietic stem cells reside and where immune cells are born. And that is a very sort of specific cluster of neurons. On the other hand, there are other clusters located in a different part of the brain which talk to muscle and which induce a different kind of shift where they mobilize different types of immune cells, in this case neutrophils, out of the bone marrow into the periphery. And these are some of the key players that I will share with you in this story. So let's go to the basics. This is the very first observation that Wolfram made, and it's a simple observation and perhaps not entirely unexpected. Here at the top, what we see is a relaxed control mouse. This is a mouse just allowed to walk around in its cage. And here we have intravital microscopy of the ear, and we're looking at cells that stain with Ly6G. So Ly6G is a marker of neutrophils. So we're specifically looking at neutrophils. And you can see there's a few cells that you can sort of see patrolling the vasculature. Those that are going through quickly, you wouldn't necessarily see. On the bottom is a mouse that has undergone restraint for 1 hour. In fact, 5 minutes is enough, provided you do this recording after 1 hour. And it seems that this is something that really mice do not like. They really do not like to be restrained. And so when they are restrained like this, for this brief period of time, and you look in their ear, and in fact, when you look in their blood, you see this massive mobilization of neutrophils. There's just tons of cells. And again, this is an observation that has been kind of made in one way or another over the years. And it was sort of interesting, and it sort of led Wolfram to think, well, let's look at this a little bit more carefully. Let's look beyond neutrophils, and let's really try to connect the dots again from what may happen in the brain as stress is perceived by these animals to what happens in the periphery. And so one way to do this was to just really do a time experiment and restrain these mice for various durations and then look what happens over the course of time. This, again, as I said, is a stress a protocol that is very stressful to the mice. The plasma corticosterone levels rise, more stressful than things like predator urine, like fox urine, for example, which was also one of the things that was tested. And when we look at this, there are really two, what I would say, distinct patterns that emerge when you look at all the major immune cell types in the body. There are, on the one hand, neutrophils, where you see this very precipitous rise in the number of neutrophils that you see within 1 hour. And then there are other cells, such as T cells, B cells, so broadly lymphocytes, as well as monocytes, where you see a decline in the number of cells that you can retrieve from the blood. And then when you let the animals recover, the patterns are also somewhat different, where on the one hand, neutrophil levels, which rose very quickly, tank very quickly. So their numbers go back to pre-stress levels fairly quickly. And we'll sort of talk about how that happens. On the other hand, when you look at monocytes and lymphocytes, it takes quite a long time for the number of these cells in the blood to sort of return to normal. And so sort of two distinct patterns, very sharp rise, very sharp fall for neutrophils, whereas for B cells, T cells, and monocytes, you have this fall and a precipitous and longer rise back. And some of the other leukocytes, the less numerous leukocytes, like basophils, for example, mast cells, follow one of these sort of patterns. So these are the sort of two patterns. And of course, that raises the question, this is the blood, of course. What happens to these cells? Are they dying? Where are they coming from? Are they coming from the lung? For example, for neutrophils, that would be a reasonable hypothesis that that they're being mobilized from some of these reservoirs in the lung, or maybe the spleen. Is there emergency hematopoiesis that is occurring here? So how do we understand this phenomenon? And so very briefly, using several adoptive transfer experiments, the bottom line is that what we think is going on is that by and large, the neutrophilia that you see in the blood is the consequence of mobilizing these neutrophils from the bone marrow. So if we account for the number of neutrophils that we can see, that we can find in the entire bone marrow, and the loss of these neutrophils from the bone marrow after stress, and then sort of do this stoichiometry, if you will, and then look how many neutrophils increased not only in the blood, but in some of these other organs like the lung, spleen, and liver, we find a pretty good correlation, which is to say that the net loss of neutrophils that we can calculate in terms of the loss throughout the various bones throughout the body and the entire bone marrow is fairly similar to the gain of neutrophils that we find in the blood and in the peripheral organs. And this was done using adoptive transfer experiments as well as just looking and enumerating endogenous cells. So this is what we think is happening with the neutrophils. On the other end of the spectrum, when we look at lymphocytes and monocytes, sort of the reverse appears to be true. So here again, what we find is that in all of these organs, skin, muscle, lymph node, liver, lung, as you can see on the right, we actually see a loss of these cells. And the only place where we don't see a loss, in fact, where we see an increase, is the bone marrow. And so that sort of, and again, this is adoptive transfer, but the endogenous populations are similar, leads us to conclude that cells are being mobilized from peripheral organs, from secondary lymphoid organs, to this primary lymphoid organ, the bone marrow, where they don't die. And that's important. And there's, in some cases, and there's evidence that, for example, after stroke, cells can die. These cells, in response to this short bouts of stress, survive, and eventually they return back. Just like when you saw that curve, eventually they return back, but it takes time. And in some cases, it takes a long time. So here we're looking at recovery in lymph nodes, and notice that even 40 hours after what was sort of just 1 hour or 4 hours of stress, After 40 hours, we still don't have the population of B cells in the lymph nodes and T cells that we had before the stress. And this has consequences on the acquisition of immunity that I will touch on briefly in a few minutes. All right, so that's the phenomenon. So this is, when we had the phenomenon, we say, okay, we see these large-scale, bidirectional leukocyte shifts in response to acute stress, and now the fun work was started, where the question was, what is responsible for these shifts? And the low-hanging fruit was, well, surely it's going to be one of two or both of these two major stress systems that we all know and love, the HPA axis and the sympathetic nervous system. There's a lot of reason to think that they're going to be important because of their roles in controlling immunity and in chronic stress and in other types of other types of issues that can arise in response to injury or infection. So we looked to see whether the HPA axis and the SNS affects these leukocytes, whether they're collaborating, and which leukocytes are affected by which. So the first thing was fairly straightforward. The HPA axis appears to be essential in really controlling lymphocyte and monocyte exit from the blood. This can be done surgically, or it can be done pharmacologically or genetically, where here surgically adrenalectomy results, really changes this, where you no longer see this decline of B cells, T cells, and monocytes. Notice, though, that now you actually have more neutrophils. So that tells us that the neutrophilia that we see under stress is not really driven by HPA axis. In fact, perhaps it is that precipitous drop of neutrophils that may be responsible, where the HPA may be important. The opposite experiment may involve the injection of corticosterone, and here we see reductions of B cells, T cells, and monocytes, which is a phenocopy of what we see in the stress. One can also do this with genetic tools. So here, specifically by knocking out the glucocorticoid receptor from B cells. So we know that this is cell intrinsic. So when the glucocorticoid receptor is deleted from B cells here, we no longer see this decline, but we still see the decline in other cells. And the same can be said for deleting the glucocorticoid receptor on T cells or on myeloid cells. So these are surgical, pharmacological, or genetic tools that, by and large, really manipulate some of the peripheral effects of HPA, but don't really give us much insight into really what happens in the brain. And so to really get and connect those dots, what Wolfram implemented in the lab was chemogenetics and optogenetics. I do not need to explain these tools to this audience, so I'm just gonna fly through them. That there are these three components that come together in terms of chemogenetics, and those are the same three components in terms of optogenetics, with the exception that in chemogenetics one delivers adenoviruses that encode DREAD receptors, and in optogenetics you're relying on channel rhodopsins. And so that's the difference between injecting a chemical to induce neuronal activation, excitation, or inhibition versus the use of light. And so one way that this can be done is, for example, by taking a CRH-CRE mouse, targeting the paraventricular hypothalamus with a surgical means and injecting an activating virus. And when you do that, we know that we have expression of those DREAD receptors because the virus also encodes mCherry. So we can see this nice stain. And 3 weeks after recovery, one can then deliver CNO and see what happens. So just to give you to show you some data with these approaches. So here is what I just explained to you. So we have this CRH-CRE mouse targeting the paraventricular hypothalamus with an activating virus. And here, after injecting CNO, we have corticosterone levels rise. And what we have is the decline of B cells, T cells, and monocytes from the blood very quickly. And in fact, we see fewer neutrophils as well. Now we can do the converse experiment by essentially deleting this area of the brain, the periventricular hypothalamus. And the mice, you can stress the mice all you want, corticosterone levels remain low, and you're unable to decrease B cells, T cells, and monocytes. One can bypass chemogenetics altogether and use the ZIM1 Cree mouse, which is essentially allows you to delete a CRH, and it's the same thing. So again, mice can be stressed, but even with these strong stressors, corticosterone levels remain low. B cells, T cells, and monocytes are not declining, which really provides you evidence that this cluster here is responsible for the shifts in monocytes, B cells, and T cells, but are not responsible for what happens in terms of neutrophils. And so, moving forward, what we would want to do is to really build the cerebral salience network and mapping input from higher fear centers, doing monosynaptic and polysynaptic retrograde tracing to really sort of build a map of what happens and how the periventricular hypothalamus receives its inputs to then instruct peripheral immunity. And so just as a summary here, and again, I didn't show you all the data, but what we think is happening is that it is the HPA axis which is driving this large-scale leukocyte shifts from the periphery, from the lymph nodes into the bone marrow, and that has major consequences on the acquisition of adaptive immunity, has major consequences on how the body is then able to acquire immunity to SARS-CoV-2 and influenza, and this was done in collaboration with Mazir Divangahi and Jeff Downey, colleagues at McGill, and Jeff is now a postdoc here. But it also has consequences on the acquisition of to autoantigens. So in the context of EAE, so the model for multiple sclerosis, what we find is that those animals that are subjected to acute stress actually are protected from multiple sclerosis effects later on. And the key here is acquisition, not exacerbation. And again, that is a conversation that we can have as to why would a system like this evolve. So the question then remains, and maybe in the last 4 minutes, so there's time for questions, how are neutrophils being controlled? Right? So we know that the HPA axis is controlling monocytes and lymphocytes. What accounts for neutrophilia? So surely this is going to be the sympathetic nervous system. We have so much evidence that the sympathetic nervous system is important. We know that under conditions of chronic stress, the sympathetic nervous system has major effects in the bone marrow. We know that by looking at tyrosine hydroxylase, which is a rate-limiting enzyme. We know that it actually induces hematopoiesis. So surely this is the answer to neutrophilia. And this is the assumption and the hypothesis that we worked on, and many months of experiments where we're sort of looking at the data scans and trying to see, well, is this statistically significant? Is this not statistically significant? Whatever we had done, and this was also done in collaboration with the late Paul Frenette, who's worked in this area for many years. It just didn't fit. The data refused to go the way that we wanted them to go, which is to say that whether we deleted the sympathetic nervous system, delivered beta blockers, did this in the context of knockouts, either beta 2, beta 3, beta 1, beta 2, adrenalectomy, or even optogenetics, no matter what we did, we could not find the SNS to be the key system that drives neutrophilia during acute stress. And so this was disappointing, but then of course the question was, well, something has to be controlling neutrophilia. What is it? And so at this point, sort of Wolfram went back to the basics and say, okay, let's think of it in a different way. Let's first ask, so if we have neutrophils that are mobilizing into the blood, something is going to mobilize them, and it's probably going to be a chemokine or some sort of cytokine. So let's measure a bunch of proteins in the blood shortly after stress and then sort of work our way backwards. And so when Wolfram did this, he noticed that there's many things that don't change at all or are not produced, but some do, one of which was CXCL1. So there's some other ones here that might be of interest, some of which we don't know what they do. But CXCL1 was particularly promising because it is a bona fide neutrophil attracting chemokine. So that was interesting because we saw that it increases in the serum, it increases transcriptionally, it increases on protein levels, and it mimics, or at least precedes, that sort of neutrophilia that we see that happens within 1 hour. And for the first time in what was months of work, what Wolfram saw was that inhibiting the receptor for CXCL1 essentially prevented neutrophilia. So it was an aha moment. Okay, so this has something to do with neutrophilia. So then, of course, the next question was, well, where is CXCL1 produced? And again, can we connect the dots to the brain? Because, of course, somehow we thought it has to be, at the end, it has to start with the brain. And so to make a long story short, what Wolfram found is that the CXCL1 is produced by a number of tissues, but it is highly produced by muscle, by skeletal muscle, which was a bit of a surprise. But it's produced there, and again, we saw that in terms of mRNA. So it's not just that it's stored, but it's actually produced. And that led to sort of going upstream and upstream and upstream to this conclusion, which I'm just going to say very quickly, is that what we believe is happening is that it is central motor circuits that control this neutrophil swarming from the bone marrow during acute stress. And this was done using a series of experiments, including optogenetics and a series of other pharmacological means. So the idea here is that the central motor circuits really induce a production of CXCL1 by skeletal muscle. And so here is some of the data involving these tools. And so here, just in conclusion, what we think is that there are these distinct areas of the brain. On the one hand, the HPA axis really mobilizes lymphocytes and monocytes out of the peripheral organs and really keeps them in the bone marrow, where they reside for a brief period. And on the other hand, central motor circuits, rather than the sympathetic nervous system, appears to be responsible for the neutrophilia that occurs shortly after. And perhaps it is then the HPA axis that dampens that neutrophil response. And again, this has consequences to infection, injury, and I'm happy to sort of discuss, how a system like this, how we think about a system like this, and what are sort of the advantages and disadvantages, and how it would have evolved. So thank you for your attention. Here are some of the people who have contributed to this work. So really, in essence, I told the story that was done by Cameron, that's the sleep work, and Wolfram, the stress work. Here are other members of the lab, many of whom have their own projects, exploring different facets of lifestyle factors on disease that I hope to discuss at some future time. And here are collaborators specific to these projects who have really sort of contributed and helped throughout these last several years. So thank you for your attention, and I'll be happy to answer some questions. And I'll be directing questions to Cameron and Wolfram here. So you guys get ready to answer. Is there anybody in the audience who has any questions?
Yeah, why don't we start with people here and then go virtual? Yeah. I don't know, which button am I supposed to push? Okay, does anybody have any questions from the audience first before you look like you have one?
Yes, what about CXCL1 exercise?
Oh, you want to take that one, Phil, and then we'll go to Alpha?
That's an online question. So which one?
CXCL1 and exercise?
and exercise. So the question is, under conditions of exercise, do we have CXCL1 levels are rising, and is that potentially contributing to neutrophils? Is that the question?
That's correct. Thank you so much.
Right.
Great
yes,
talk.
thank you. So this, there's a sort of a deeper question in this, in this simple question, and that is, well, if motor circuits are sort of inducing this neutrophilia, motor is responsible for movement, right? So what happens in exercise? And so the key here is whether it is voluntary versus forced exercise. So as it happens, if you, for example, allow mice to run on a treadmill, they will run something like 10 kilometers in a 24-hour period. So they love to run and they love to exercise. That kind of exercise does not induce neutrophilia. And actually, when we looked at just the motor movement and just the intensity of the muscle twitch, if you will, under conditions of voluntary exercise versus under these conditions of stress, there's a big difference, right? So you just do not. So we think it has something to do with just the intensity and the frequency of the action potential that you're inducing under these conditions of fight or flight or these fear of death situations. And so you just do not cross that threshold. And despite the fact that these mice, when they're moving and running on their wheels, that their muscles are engaged, it's not sufficient to induce that kind of response and that kind of neutrophilia. Maybe very different if you're electrically shocking these animals to exercise with intensity, but of course shocking induces stress, so that's a difficult one to test, right? But it does raise a question about what happens when we run, whether we run a marathon or a sprint, and certainly when we do this, you can, I invite you all to collect your blood after a run and count your monocytes and neutrophils, and you will see that these numbers change fairly quickly.
Well, make sure you donate to the lab so that we can actually.
Yeah, exactly. Yeah, that's right. Just don't. Yeah, come back. That's right.
Amazing work. My question was related to the stress-related work. So for the acute stress, these are the changes you show, but I was wondering about chronic stress. Like for the chronic stress, is that something you're looking in the lab? And then also, like, would you expect, like, acutely when the chronic stress starts, you'd see this pattern, but then it would be quite different or worsened?
Yeah, yeah, this is another very big question, and so in order to answer it, one must sort of take you sort of consider a few sort of key issues here. One is, how do we go from acute to chronic? When does repetitive acute stress become chronic stress? Well, it's, you're repeating it, right? And then acute stress, of course. And certainly in the mouse, there are different types of chronic stressors. There's this chronic stress called the chronic variable stress. There's the chronic social defeat stress that Scott has really pioneered. And those different types of stressors really induce different biologies. What we're finding is that there is this sort of issue of susceptibility and resilience. So when you look at these phenomena that occur very acutely, as you're repeating the stressors or as you're putting animals in different types of stress situations, certain phenomena repeat themselves, or at least are not trainable or adaptable, while others can sort of adapt or change. We don't understand how that happens, but we think it's very much a key to this concept of resilience or stress inoculation that I think is very much sort of an area of investigation that I think is fascinating. Certainly under conditions of chronic social defeat stress or chronic variable stress, the sympathetic nervous system kicks in as important and induces hematopoiesis. We don't see hematopoiesis in these acute situations here. So I think there's a lot of moving parts. So it's not easy to answer the question sort of one way or another. It's just that is that there I guess one lesson is that the body adapts. There's no question, but it adapts in different ways that may mean different things for different leukocytes.
Hey, Phil. So coming back to the sleep part of the story, so you disrupt sleep and then you mobilize more monocytes. And if you have atherosclerosis, that's bad. But if you're young and you don't have atherosclerosis, what else is going on there with those monocytes? If you could trace the fate of those cells versus, you know, what would you expect to see?
Yeah. So that's interesting, you know, because we are looking at this in the context of a disease, right? So we have hypercholesterolemia, and of course one can say, well, if there is no disease, it's okay to have more monocytes, right? On the other hand, you could say, well, do you really want to have monocytosis? You know, monocytes circulate in the blood. They follow a circadian pattern, and they have, you know, a great sort of consequence on response to injury, infection. And one argument is that we're never really, truly, quote unquote, healthy. You know, homeostasis is maybe a misnomer. There's no stasis. There is like homeodynamism, right? But if you're 40, 90% of us who are 40 already have pretty advanced atherosclerotic lesions. And unless your LDL cholesterol is under 70, you're probably gonna have atherosclerotic lesions. So by and large, I would say that you probably don't want to have monocytosis unless you really need those monocytes to fight an infection or to fight injury. And just from a more philosophical perspective, what we're seeing with this disruption of sleep is really a disruption of a balance, right? By reducing hypocretin levels, by essentially taking those breaks away, we are seeing this acceleration of hematopoiesis, which has consequences on clonal hematopoiesis, on the diversity of the hematopoietic pool. That's something that is in the study that is under revision. And so we're sort of needlessly, if you will, accelerating a system through just by not sleeping well enough, right? So the system is resilient and flexible, but by essentially cutting on our sleep, we're kind of like pushing it more than it should be pushed. So that's kind of ways of thinking about it.
I'm going to switch over to online. I'll just repeat them for you. So the first question online is from Mel Berkowitz, who wants to know whether taking a nap in the late afternoon reduces atherosclerosis.
I wish. So there is a lot of literature on this, growing literature, really good literature. There's this concept of chronotype. So I'll get to the answer, but just by way of introduction. So everybody, each one of us has their own chronotype, which is your sleep need. You know, some of us need more sleep than others. On average, we all fit in that bell curve. We need between 7 and 9 hours. Some need more, some need less. And paying attention to your chronotype is very important. So that's one. Two, if there is sort of one rule of thumb, it's about regularity, right? So catch-up sleep on the weekends doesn't really work. There's studies that looked at this. Like if you have a sleep debt from Monday to Friday, sleeping as much as possible on the weekend just does not provide you with a health benefit. So it's about sort of finding regularity. So if your regularity, you know, if you're like Kramer, Kramer from Seinfeld in one episode was like, I'm going to sleep every 20 minutes for 15 minutes or so. Like if you find regularity through having a nap, perhaps, you know, that might be helpful. But it is the disruption of that rhythm that seems to be specifically harmful, and also deviation from your chronotype. So I'm actually considering getting that Oura ring to actually, I know when I slept well. I know how I feel when I slept well, but those who have the Oura ring tell me that it's actually a really great way to really sort of correspond how you feel about your sleep to what the data tell you because Oura Ring is not the only one that tracks your sleep, but apparently it's quite effective and has helped people sort of improve their sleep hygiene, if you will.
So we're after the hour. I don't know if people want to hang out. There's still some more questions online. I'll keep firing them away if you're game.
I'm fine, or they
right.
can also be brought, emailed. So maybe one or two.
Let's do one more. And then if anybody else has a question in person, we can go to that for the last wrap up. But let's do this one from Yajing Zhu. Thanks for the talk. I also read your paper with great interest. I have two questions. One is there any link between the HPA axis induced lymphocyte aggression and the muscle induced neutrophilia? Are they independent? Two, under what circumstances of normal physiology outside of restraint stress would you get equivalent muscle activity to induce CXCL1 and neutrophilia?
Okay, so let me answer the second question first. What we think is happening is that only extremely life or death situations induce this. So Wolfram had compared different types of stressors, and I alluded to that during the talk, that, for example, fox urine doesn't cut it, right? So, but maybe interaction with a very aggressive mouse will. So this is a fight, flight, or freeze phenomenon. And it's a phenomenon that is essential to our survival, you can sort of imagine, right? So, you're hanging out and there's a predator that is going to chase you, 10,000 years ago, or if you're, at a safari without some safety precautions right now, it helps to have that mobilization, and it helps to have that sort of extra push, and here neutrophils may be really responding in this phenomenon of anticipatory inflammation, where they're going to locations where they may be needed most in case there is an injury or infection. So the second question as to the connection between the HPA axis and the motor circuits is very important and interesting, because on the one hand, I spoke about these distinct regions, like there's this little cluster neurons, there's a little cluster of neurons, but we clearly know that there is connections, that the brain is talking to itself all the time, and there are monosynaptic and polysynaptic connections, and so this is very much something that we are interested. I showed that one slide with the retrograde tracing techniques because these innovations occur, and I think this is going to be something that is going to be a focus of work to come.
Fantastic.
Okay.
I think we should probably end here. Let's give a huge round of applause to Phil for an amazing talk. You really raised the bar, and I should note there was 120 people, and you've maintained 72 people online even 10 minutes after the time is up. So please join us. We're going to do this every month. Join us next month. We've got another great speaker, homegrown great speaker, Brian Kim, talking about the neuroimmunology of itch. So thank you very much.
Thank you, everyone.
Day.
Thank you.